Hi everyone, welcome to Unit 2, which will cover microbial diversity. During Unit 2, we'll learn more about prokaryotes, eukaryotes, and the acellular pathogens that create the microbial world. Let's start with prokaryotes. Prokaryotes are ubiquitous, which means they're found everywhere on the planet, even in the atmosphere. There may be up to 2,000 different kinds of bacteria in the air. Prokaryotes are also adaptable, which means they're metabolically flexible. They can switch from one energy source to another depending on resource availability. As we know from our early studies on the microbiome with the research project, we know that prokaryotes, like bacteria, are abundant on the human body. Scientists estimate that bacterial cells outnumber human cells 3 to 1, maybe even 1 to 1 by some studies. And finally, prokaryotes are important for the health of our ecosystem. They're a necessary part of soil formation, and they work hard to keep our soil and air clean. Prokaryotes capture and recycle elements like carbon and nitrogen. We call this fixing. Animals like humans require organic carbon to grow and cannot use inorganic carbon sources like carbon dioxide. Because of this, animals rely on prokaryotes to convert carbon dioxide into carbon products that they can use. We call this carbon fixing. Prokaryotes can also convert atmospheric nitrogen to ammonia, which is important for plant metabolism. Without this collaboration, it would be impossible for humans and plants to survive on Earth. These abilities to fix inorganic substances is also very useful for keeping the environment clean. Some bacteria can degrade toxic chemicals that can pollute water and soil. When bacteria metabolize polluting chemicals to reduce toxicity of soil and water, we call that bioremediation. You may have heard about this with some efforts to use bacteria to reduce microplastics in the ocean. A friend of mine from graduate school studies sites where companies dispose of old paint and is systematically characterizing the natural soil bacteria that seem to be able to clean the paint chemicals out of the soil. The second group of prokaryotes is archaea. Just as bacteria were good at fixing carbon, archaea can also contribute to recycling elements. Archaea grow in extreme temperatures and conditions like the hot vents at the bottom of the sea or the nutrient-dead Dead Sea. We also see lots of archaea around the natural geysers and hot springs. Some archaea are methanogens, which means that they can break down carbon dioxide if hydrogen is present. So we'll learn more about prokaryotes in our activities that are geared for Chapter 4. And then in Chapter 5, we'll learn about the 5 types of eukaryotic microbes. Those are parasites, fungi, algae, and lichens. We'll finish Unit 2 in Chapter 6, where we'll learn about acellular pathogens, viruses, viroids, virusoids, prions, and bacteriophages. Despite all the positive characteristics of prokaryotes that I've recently described, sometimes they can make us sick. Microorganisms that cause illnesses are called pathogens. Pathogens are responsible for illnesses in a number of organisms, plants, animals, and bacteria. They also contribute to food spoilage and foodborne illnesses. And as a person who loves street food, I am more than familiar with this characteristic of microbes. Prokaryotes also play a role in climate change. As glaciers melt and sea levels rise, soil that was previously frozen called permafrost is warming and causing a reawakening of bacteria that is releasing massive amounts of carbon dioxide and methane byproducts into the atmosphere, and this is contributing negatively to the greenhouse effect. Okay, now it's time to look at the Unit 2 activity map and see what we have planned for this unit. So I'm trying something different, so you'll see some notations on each of the little post-its. The IV stands for the Play Pause It Interactive Video. Where you see R and A, that means you'll do a short reading and answer a couple of questions. And where you see R and D, you'll do a short reading followed by an activity. So the first activity is one of those Play Pause It interactive videos. So we'll learn about gram staining and then do a couple of answer a couple of questions to test your knowledge on that. And then we'll move on to a prokaryotic diversity assignment. So you'll do a short reading. This is all in Blackboard, and then answer 2 questions. And after that, we'll move to number 3, which this work is folded into our collaboration with TCC. So you will work through Chapter 4 on your own. You'll pick a bacteria to profile. You'll look it up in the chapter, and write a little profile, and we'll turn that into a case study. So this will also be work featured at the study table on Thursday if you want to save that for group work. And then for the fourth item, we'll walk through chapter 5 in our eukaryotic diversity activity. So this is, you'll do a short reading and then an activity after that. The fifth item is we'll learn how to identify bacterial cells, types of bacteria, by their shape. So that's a little read and do. And then the sixth activity is an interactive video on the viral life cycle. And then we'll finish up the week with a Unit 2 assessment. You see the seventh item is our research project, so that will post right at the top with everything else, so you can scaffold that work through the week. And we'll also, if you need help with that, there'll also be time at the study table to work through the research project. And just a couple of final reminders before we go. Don't forget to check Blackboard. Look in now, see your grades, see what was posted for last week, make sure everything looks the way you expect it to look. Also, if you haven't already done so, register for the interactive labs at Pivot Interactives, because we're going to use those next week. So the class key is here. And then finally, as always, drop-in office hours are Monday through Thursday, 10 to 11 a.m. And again, I'll have the live study table on Thursdays from 3 to 5 p.m. So all of this information is posted on the Blackboard course shell. So enjoy Unit 2, and I'll be around if you need me. Good luck.